Hi everyone, my name is José Alexandre Diniz-Filho, and it's a pleasure to welcome you all to another event organized to celebrate Humboldt's Day, organized by the International Biogeography Society, the IBS. And I would like to thank to Miguel Matias, Sandra Nogueira, Karen Fowler for inviting us to participate in this event and for all the support from the IBS. So let me invite my colleague here in the organization of this symposium on local process and emergent patterns in neotropical biogeography, Dr. Elisa Barreto. Elisa is working with Catherine Graham in the WLSL in Switzerland and working here with us in Guyana too. So welcome, Elisa. Hi.
Hi everyone. So my name is Elisa. I'll be co-mediating this section with José Alexandre. In this session, we will have the pleasure of hearing from three great researchers from different geographic regions and biomes in Brazil that are working on different research fronts to understand local processes and emergent patterns in neotropical biogeography. And let me introduce you to them. First, we will have Dr. Sid Gouveia, who's a professor at the Federal University of Sergipe in northeastern Brazil. He studies multiple aspects of macroecology, biogeography, and biodiversity conservation. Sidney will kickstart our session by talking about mechanistic models for phenotypic diversity. And welcome, Sidney. Thank you for joining us.
Thank you, Elisa, José Alexandre. Thank you very much. It's a pleasure celebrating this the work of Humboldt, right?
Right. Thank you. Next, we will have Dr. Fernanda Werneck, who's a researcher at the National Amazonian Research Institute. Her main lines of research include evolution, systematics, biogeography, and conservation of neotropical herpetofauna. Fernanda is also engaged in projects to promote and value women in science, and I'm sure she will be happy to address questions related to this as well. So thank you very much, Fernanda.
Hi, thank you. Thank everyone. Good afternoon. It's really a pleasure to be here and hear you all.
And lastly, we will have Dr. Thiago Rangel, who's a professor at the Federal University of Goiás in central Brazil. He has broad experience in ecology, statistics, and computer science. Thiago has been studying the drivers of organic and cultural evolution at large spatial and temporal scales. And he is especially inclined to using simulation models to address ecology and evolution of biodiversity. And he's going to talk about this today. Thank you for joining us, Thiago.
Thank you very, very much for the invitation. It's a great honor to be here to honor one of my greatest academic heroes, von Humboldt. He's an inspiration for all of us, and we are all walking the path that he has started. So it's an honor to be here and to share with you the academic work that I've been doing with my research group.
Thank you. So before we start, let me quickly explain the dynamic. Any questions or comments you might have can be made in the comments section of YouTube or Facebook at any time, and they will be addressed by the speakers by the end of all three presentations. And then we can have a broader debate with all of the participants. So please don't be shy. Leave your questions and comments. Well, I think that's it, José Alexandre.
Yeah, we'll have the three presentations, about 20 minutes each one, and then we can move to the questions, and you can write the questions, and then you can we will transfer the questions to the main screen so everybody can see the questions, right? So I think we can start, right? We will start with Sidney, and then we'll go to Fernanda, and then to Thiago. Okay, so I'm going to put everybody in backstage, and we can share the screen by Sydney, right? Here we go, Sid. Okay, it's online. So I'll be in the backstage if you need something, right? And thanks again for accepting our invitation. It will be a pleasure to listen to your talk. Okay, I'll be here.
Okay, thank you, Alexandre. It's a great pleasure to be here. It's an honor, actually, being talking in this event that celebrates the work of Humboldt. I think we own him a lot because everything we do. So I'm here to speak a little bit of mechanistic model for phenotypic diversity, and to kick off, far from creativity, let me start with a quote of Humboldt, in which he say that the nearer we approach the tropics, the greater the increase in the variety of structure, grace of form, and mixture of colors, and also in perpetual youth and vigor of current life. So the Neotropics was, I think, the trigger for all the thrill for Humboldt, for Darwin and Wallace in pursuing this understanding of biodiversity. So in addition to own Humboldt, all of this, we own the Neotropics, all of this. So Humboldt pioneered a view or approach to understanding biodiversity that involves trying to understand ecological mechanisms or rules of biological diversity. But besides Humboldt, some decades later, another persona that was very important for the biogeography was Wallace, which introduced, or was the main one to introduce a perspective, a historical perspective, or trying to understand biodiversity from its historical background or context. Nowadays, we see the biogeography and other fields such as macroecology and theoretical ecology trying to integrate these two perspectives of the understanding of biodiversity. One of the greatest ideas of this is the idea of the evolutionary play and the ecological stage, and where all these dynamics happen. So what I'm trying to show here is actually how we can search or pursue this integration of rules in history of these ecological mechanisms and the historical context, and trying to understand the diversity, and specifically diversity of body size diversity. So just a quick introduction for the earlier the younger biogeographer. We measure diversity based on the quantity of types of things. Things can be species, genes, traits, or lineages, and how the many things of this we have, the more diverse is a region. But also based on the frequency of these types of these things, such that when we have a more even distribution of these entities, we have a higher diversity. And why I'm saying this, why I'm showing this, because this is the basic framework which I'm trying to introduce the idea of diversity and discuss the diversity of body size in this presentation, this work. So I'm trying to look at body size diversity in this based on this framework of frequency distribution of size classes. And of course, I need to tell you what exactly I'm I'm studying, what biological group I'm studying. And I'm talking about the size diversity in South American ground lizards, which has a huge diversity from very, very small, I don't know if you see my cursor here, very small lizards such as this Paranaguan ground gecko to this huge red tegu. I don't think this scale is proper. Maybe if I put it in right scale, the ground gecko would be too small to see. So this is what I'm working on right now and what I'm trying to show you. So as I said, we're trying to integrate the rule, the idea of developing a mechanistic understanding of the process of the patterns, the biogeographic patterns, and this rule is based on this model that was present in two related studies led by Juan Rubalcaba, in which we proposed them. They are a little bit different, but basically the idea, the fundamental idea, similar, in which we apply to try to understand this trait diversity, or in this case, the size diversity of lizard, of ground lizard. Okay, and what does the model do exactly? The model integrates biophysical and physiological functions into hypothetical populations, and how these properties emerge to macroecological scales. And from this we construct macroecological indicators of the processes we are investigating. This is the basic idea of the model. I'm showing it in a bit more details in a minute. But the basic questions of what we were studying was to what extent biophysical, in particular thermal and hydric constraints, determine the size diversity of tropical lizards. And we asked also if those historical background, specifically past climates, relate to the model mismatches. So when model failed to predict the size diversity, we thought that we may be it would be related to historical factor, recent historical factor, in this case past climates. Okay, so the model builds a computer lizard, a lizard that reacts to the environment, and this is some basic characteristics of our computer lizard. It's a diurnal ground lizard. It's a ground lizard. This is important to point out because the model is suited to a ground lizard. It operates in the microclimate that happens near the ground. It doesn't work well for boreal lizards, for instance. This lizard has a typical body shape, head, tail, and legs, and based on known allometry relationships. It has a preferred body temperature of 27 degrees, which is the average of some lizards in South America. He has a typical thermal asymmetric performance curve. He has a behavioral thermoregulation. This is important for the model. And he has a color. Yes, he has a color. It has a higher emissivity to long-wave radiation, which means that it's close to really dark. So I like to think that it is dark gray. Okay, so this is a very it's a complex model in the sense that integrates several biophysical, physiological functions, but it is simple because it's the lizard is homogeneous. It's equal for everywhere. The only thing that vary in this lizard is its size. Okay, it varies in between 0.1 gram to 3,000 grams, to 3 kilos. Okay, because we are particularly interested in this understanding the pattern of diversity of body cells, of course. So we need to keep everything else as constant. What the model does is to integrate in the body of the lizards the physical processes they operate in the environment. Radiation in, radiation out, emitted from the body, convection, evaporation, conduction, and these all raw physical processes, they determine the quantity of heat or the variation of heat in the body of the lizard. And this variation of heat determine the temperature, the body temperature of these animals, and this is related to its mass, to the size. So this is the particular aspect that makes all these processes to vary among different animals overall. Okay, in this case, this lizard change how they integrate the environmental conditions in the body. All these functions are scalar functions, actually. Okay, so this is an idea how the model works. We have a thermal landscape that's changing in a minute basis, and at each moment the lizards needs to decide what to do. And it's not a individual-based model. I will explain it in a minute. But the model, the animals needs to decide what to do. If it stays, if it moves, if it changes the posture, body posture to preserve heat, and these decisions of the lizards are decoded as probabilities, which depends on the temperature and the particular environment where it is, their preferred temperature, the optimum temperature, its thermoregulatory ability or behavior, the temperature of the next, the spot next to it. So this is decision lizard has to make to move to optimize its body temperature. Trying to predict what a particular single lizard would do in this situation, it's extremely hard. Trying to predict what an entire population of lizards would do is astronomically harder. So how do we? In fact, it's a lot of lizards thinking of probability. It wouldn't work. So how do we handle this huge complexity of information? For this, we employ the maximum entropy principle, in which we treat each individual as a particle. In this case, it's a living particle. We add a living aspect or living trait in the particle by informing that the animal has to optimize its body temperature based on the performance curve on its preferred body temperature. So what the maximum entropy principle says, it says that any physical system tend to uniform distribution, or the most uniform distribution given the constraints. These constraints, in our case, are microclimates or the available microclimates, the thermoregulation of the animals, and the biophysical processes they are solved on a minute basis as he spend the time in the particular environment. And this will give us a distribution probability, a maximum entropy distribution probability, which says what is the final or the operative temperature, this temperature that the animal ends up with. And with this temperature, we can compare it with the performance curve of that species, the particular size class, and from this we can measure an accuracy, a thermal accuracy, which tell us how suitable that climate is for that particular size. Okay, and with this measure of accuracy, we can map this accuracy and interpret the pattern, geographical patterns of accuracy, and how this particular climate fits, or the size fits the climate. Okay, so these are the maps we have with this measure of accuracy of divergence between of how close actually the size is for that climate in terms of biophysical exchange of heat with the environment. When you first see this maps, you can say, well, yeah, it's pretty good for any lizard in South America. It's warm and nice and everywhere, nearly everywhere. So it's pretty good for any lizard. So the best way to check how good this model is to compare, of course, with real data. And this of the distribution of each size class of lizards, of ground lizards in South America. At first, it's a lot of information. We can see pretty well if it's good or not. We can see some really interesting associations. In this case, for the very small lizards, it's smaller than 1 gram, and the others are okay, pretty good. So how we measure this correspondence? We measure this correspondence, and they are fairly good. Okay, we have a fair correspondence between this model. One important thing: the model is not suited to describe diversity or richness of particular sizes. It is designed to inform if a particular climate fits or not a size. So it's a yes or no question. What we are doing here is a bit further, asking if this accuracy, how good the environment is, can predict the diversity of particular size classes, and it works fairly well. Okay, and because we are working with distribution, we measure the entropy of the distribution, distribution about the size and the accuracy through the Shannon-Wiener index of diversity, and this is what we got. Maybe not that good, but the overall trend of diversity of the distribution of frequencies of different sizes has a fairly good match. You can predict, for instance, this sudden decline of species with different sizes below the crabcorn tropic. Of course, we have some more species here, but species doesn't always obey our rules, right? Overall, we had a fairly good correspondence. Remember that the model was not properly designed for that, but still we have a quite good correspondence between the two models. The second question was, we had this hypothesis beforehand, was that this divergence between these curves was related to recent climatic changes. And the most significant climatic changes we had in South America was the change from glacier, from the glacier maximum to the current climate. So we try to compare this relationship between the divergence, the diversity, the model in the real data and the empirical data with climate changes since the last glacial maximum. But no, it didn't work well. So at first, there's something else that is behind this divergence. Maybe the model is not that good, actually, obviously. But we thought maybe since the size itself are very specific in the response to climate, maybe there are some differences in or size dependency in this association with the change in climate since the last glacial maximum. So we analyze this divergence based on the each size class, and this is what we got. It improves a little bit the explanation of this divergence between the model and the data. It's more important for some size class than others, but overall was a weak effect of recent climates. Apparently, of course, the model worked pretty well in predicting an overall general pattern, but its divergence to the real data was not very well explained by this particular hypothesis we had. And from now on, what I'm doing is pretty much a posteriori speculation. Okay, just because we have in this group of brilliant speakers, Fernanda Werneck, who is an expert of lizards and South American lizards, and in particular, she may help me out with this part. But I think, or I speculate, based on these results, is that we have a consistency in most size classes of a region where it appears to be more divergent regarding the model as compared to the model. And these regions may be related, this is speculation, to stable warm open areas in South America. And Fernanda may help me out with this. And that's it. I think that's it. My time is up. Just to conclude, from our results, we think that size diversity of South American ground lizards can be fairly explained by heat balance. We knew in first hand that the model was not designed for that particularly, but still it has a good fit to the real, to the predictor, to the empirical data. The effect of historical context was low in this case, was not really strong, but it appears to have seems to be a dependence of size. So different size may have different responses. In fact, different sizes are distributed differently in the space, and was distributed differently in space. So how each size responded to past climate was actually, or was probably different. And this has a huge, or that may have some influence in the current model. Okay, and this last, as I said, pure speculation, the discrepancy between actual and predicted diversity may be suggestive of relevance of prior diversifications based on this vast literature that Fernanda Iguarino has worked about lizards from South America, in which they show that there have been a lot of specific cases of lineages with high diversification in some particular areas, and this may be important, or this may be the historical context that I told that matter, or I told that is critical for understanding these two aspects: the rule, the ecological rule, and the historical background or context. Okay, that's it. I'd like to thank this great Smiley team: Elivelto Nascimento, Thalita Amado, Paulo Martinez, and Juan Rubalcaba, funding, and you all for listening. Thank you.
Thank you, Sid, for the talk. Fantastic. It's amazing, this model that you guys developed. Very, 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 very nice. Thank you. And I'm going to put you in the backstage so we can invite our next speaker, and we'll discuss all the integrated issues about local process and broad-scale patterns. In the end, I think that your presentation shows very clearly the overall idea of the symposium that I did not talk in advance, that is to integrate what is happening in very local scale, even within the individuals, up to the macro scale, that we see the emergent patterns, that we can discuss all this relationship from different points of view. So your model is very, very, very interesting in the sense, and that's why we invited you for this overall idea. Thank you. Well, so it's a pleasure to introduce again Fernanda Werneck for her talk. Hi, Fernanda.
Hello, hi. Hi, Alexandre.
You can share your screen.
All right. Just a minute. Is it up?
Here we go.
José?
Here we go. Yeah. Okay.
Can you see the screen and
Yes.
everything else? All right.
Yes.
Okay.
So
Thank you very much.
You must select your talk, right?
Sorry?
You must select your screen. You're sharing the entire screen, so you must put it in the PowerPoint.
But can't you see right now the share, the
We
screen
can see
share?
we are seeing all the studio, right? So you must.
Oh, so I selected the wrong one. Sorry. Let me stop. I select the wrong screen.
Let me remove. Okay, go again. You have to share the window or the entire screen. So then you can move to.
What about now?
We're still in the same.
So you are seeing the
studio.
Okay. So it's screen number 2. Okay. What about now?
No, it's the same.
No? Oh, that's weird because I changed the.
That's. Let's try again. Why don't you interrupt the exchange?
I did. Then screen. You are sharing the wrong screen, but I changed it. I tried 2 of them. Let's see again. What about now?
No, it's the same. Let me remove and go back. But it's the same. It's the same screen, Fernanda. You can share or the screen or the particular window of the PowerPoint, right? You must put the presentation.
Yeah, we did that yesterday, and it worked pretty well.
Yeah.
I'm on screen 1. I moved the presentation here. So can you see the presentation?
No.
Now?
No, it's the same.
Okay, so instead of sharing the screen, I will try to share the window. Let's see if it changed. So, but if I do that, what are you seeing? Are you seeing the talk?
Yeah, if it's okay, yes, as we did before.
Okay, so os slides estão passando, Diniz?
Not yet.
So that's the deal. If we share just I will switch to Portuguese just so we can figure this out. Se eu compartilho só a tela, ela não passa o slide. Você está vendo a entrada principal aqui? Sim.
Aí você tem que dar um Alt Tab e ir lá para sua janela, né?
E agora? Não, né?
Agora sim, ok. Eu vou adicionar.
Mas calma aí, você viu passando o slide ou não?
Agora tá ok.
Mas está aparecendo as notas ou está aparecendo o full screen?
Tá aparecendo, você está compartilhando o PowerPoint, né? Você teria que agora você pode botar a apresentação.
Não, mas tá, a apresentação tá aqui para mim, ela tá no Play, Keynote, Live, tudo. Vou tentar só mais uma vez a tela, só um minutinho. Que estranho, né? Porque aquele dia a gente fez, eu já fiz outras vezes isso.
Tranquilo, sem
tela, allow. E agora?
Agora sim, ok.
Mas você está vendo the full screen or the presenter notes as well?
Continua errado. Ele está agora mostrando a sua visão do PowerPoint. Você
então
está vendo
com o presenter todo. E agora?
Agora sim, ok.
Tá, passou, mas did the slides went through?
Yeah.
Tá passando? Sim?
Passando.
Ok. All right. Okay. Sorry about that.
No problem. Thank you, Fernanda.
Okay, and do you see the bar that Sid had on his screen, or it's off?
No.
Okay, all right. Okay, sorry about that. Thank you, everyone. Good afternoon, everyone. And I would like to start off thanking IBS and Zé Diniz-Filho for inviting me to be here today. It's a really pleasure and honor to talk on the Humboldt Day and honor his legacy. For such an important researcher for biodiversity research, right? So today I'm going to talk a little bit about climate change impacts on the neotropical biodiversity. And of course, it's no news to everyone that our Earth house is literally on fire, and that global climate change is a reality that challenges biodiversity and actually all forms of life, including ours. And we definitely need to learn how to deal with it. However, even though society perception on climate change is improving and it's growing a lot, some very recent studies, they indicate that media coverage, for example, of climate change, it's up to 8 times higher or greater than coverage of biodiversity. So in other words, even though the society somehow it's learning more about climate change and know that it's happening, it has very little idea of the consequences of climate change for biodiversity. So the links actually need to be made and bring to the public awareness. And in fact, global biodiversity is going through a very serious crisis. So according to last year numbers from the IPBES report, up to 25% of species, they are under threat, and more than 1 million can be considered under imminent extinction risk. And the main threats and the main causes of that are the effects or the combined effects of climate change, land use, and also other threats for sure. So the Neotropical region, although having this amazing biodiversity and the highest levels on Earth, that amazed several researchers for ages, including Humboldt, the Neotropical region, it's being hit particularly hard. So very recent studies, they show that the 2 largest Neotropical biomes, the Amazon rainforest and the Cerrado savanna, they are very close to the tipping point, even in terms of species extinction in the Cerrado and also of non-forest conversion when we talk about the Amazon. So, and these threats, they can be actually even stronger in some regions. So, for example, historically, the deforestation of the Amazon, it has been concentrated along the arc of deforestation, which largely coincides with the natural transition zone between the Amazon and the Cerrado, or what I'm calling here the Amazon Cerrado ecotone, or the gradient among these 2 biomes. And as most of biomes across the world, this region has been historically neglected in terms of conservation efforts and scientific studies. So it's a particular focus of my research group. So here today, I want to share a little bit with you some of the approaches that my research group is using to answer a fundamental yet very essential question, which is if species and their natural populations from the mega diverse Neotropical region, more specifically the Amazon Cerrado gradient, if they can escape extinction from climate change through the use, let's say, of adaptive processes. So more specifically, we focus on amphibians and reptiles, first because they are awesome and beautiful, as you can see, but also because they are ectothermics, so they depend on the environmental temperatures to control their body temperatures, and they have some pretty strong associations with local environments and climates. So we expect them to be under very high threat from climate change. So in general, the species, they can have two sorts of natural responses to climate change. So the first would be go through some distributional shifts and then track down the favorable climates across space and time. Or they can go through some adjustments to these new conditions, the new environments, either through phenotypic plasticity or adaptation, genetic adaptation. But if they fail to have one of these responses, they might go through some demographic collapses that can ultimately cause extinctions, both at the local populational level, but also across some entire species range. So that's known in some cases. So, but to have any of these sort of responses that I mentioned, they need to have a number of conditions, let's say. So they need to have enough time to move or adapt, adjust, right? They need to have the dispersive capacities and also the genetic diversity to support an array of potential responses. And that's usually not the case because climate change way too fast, or species don't have the capacity to track the change or the dispersive to move in space. And usually the adaptive genetic variation, it's not enough or is not considered in these studies. And that's the case for the Neotropical region. So we know very little about adaptive genetic diversity, especially across the space. So if we want to look at these possible responses, there are a number of ways that we can quantify, a number of methods that we can use to do that. And for quantifying range shifts, the ecological niche models, or the ENMs, they are the most broadly used. And its most simple application, which is called the correlative ENMs, the current species distributions, they are the latitudinal and longitudinal data. It's used as a proxy to understand the set of favorable conditions for species occurrence, and then this can be projected back in space and across time periods as well. So for example, I bring one study from my group in which we were looking at 26 species of venom snakes from the viperid family. And when we superimposed the suitabilities across time scale, we have found that even though richness, like the total richness across the South America, is not expected to change a lot, we saw that it's expected to have a lot of turnover of species in future scenarios of climate change. And we found also very similar results for lizards and also for other groups such as birds in the Cerrado savannas, for example. But this approach has some potential limitations. First, it assumes equilibrium with current climates and the idea of conservatism of this equilibrium across space and time. So there is a different set of class of ENMs, which are the mechanistic models, which Sid explained it wonderfully. So I won't go into too much details, but the idea is that instead of only occurrence data, they also will use some biophysical properties of the species collected usually experimentally, for example, physiological data, as a better proxy or a functional link between species properties and their the environmental conditions that are favorable for these functionals, let's say. So, and these can also be integrated in ENMs and be projected back in the space and time as well. So, as an example, I bring here some studies from my group in which we were looking at the Amazon Cerrado gradient lizards. And here is a species of forest-associated lizard. And we found a lot of intra-specific or population level variation in the traits, in the physiology traits, and also in the vulnerability. So some regions some populations seem to be way more vulnerable and be under higher extinction risks than others. And we also found that along the Amazon Cerrado ecotone, some of the traits, they are very different. So very higher thermal maximum or critical maximum temperatures that these populations can face. So that brings us the idea that maybe these populations that are along the ecotone or the transition between these biomes, they might have been under historically stronger selective pressures from these harsher environmental conditions and likely higher temperatures for these species that is mostly distributed in the Amazon basin. And maybe they have gone through some sort of climate-driven adaptation. So with that, we have this like a more recent approach to infer the ENMs that incorporates the possibility of adaptation within the framework, so adaptive landscapes through the use of some population metrics or mechanisms, along what they said. So here I have we have with us the authors of the study. So the idea is that if we look at the trailing edge or the edgy limits of the potential distribution that are actually very different from the current conditions, and in the future might not be favorable for the species. So if we look at things that are happening here, so for example, maybe some of these populations are pre-adapted for these bordering or harsher conditions, then we could incorporate some parameters of evolutionary rates, and this be taken into account in the ENMs. Even if it's a theoretical framework, it takes into account the possibility of adaptation at these regions. But we can also take that possibility into account by looking at the genetic level information that species and populations can bring us. So for quantifying the other sorts of responses, the adjustments responses, there are also, as for the ENMs, there are several approaches. And basically, so the approach I will speak in a minute, but the two sorts of responses, they can be either plasticity, which is usually determined by the environment, and it's not a multi-generational, while evolutionary adaptation has a selection basis on environmental selection, and has a multi-generation basis. So we could maybe look at the standing variation today, and it might reflect adaptive processes that happen in other generations of like heat adaptation and cold adaptation, for example. So we could investigate these possibilities using different approaches, genomic level approaches. So one of the most used approach or commonly used approach are the so-called common garden experiments. So in this sort of study, usually genetically distinct populations, they are compared under identical controlled environmental conditions. And then it makes easier to tear apart the effects of plasticity and adaptation. But of course, that's a challenge for several groups, and especially natural populations of non-model organisms that you just can't bring in the lab and grow them in the common garden. So another approach would be the transcriptomics, in which gene expression can be quantified under different environmental treatments, such as increased temperatures, and then a differential expression might be observed under these treatments. And finally, more thanks to the high throughput techniques, sequencing techniques, we can also identify signatures of local adaptations by relating the genetic diversity with the environmental variation across these populations distributions, such as here. And this can be done through analysis such as genome scans or the so-called genome environmental association studies. So in that case, we are looking for loci or SNPs that are very likely under selection by environmental variation. And we can look at this and see if this variation represents a substantial variation to look at evolutionary rescue potential. So the underlying idea is that if we focus on the standing genetic variation that evolved through climate mediated selection pressures, we would have a good idea of the adaptations that are currently in place, but have evolved by climate selection. And these, the populations that have this sort of standing genetic variation, they could be potential sources of evolutionary rescue for populations that don't carry this standing genetic variation in the future, right? In scenarios of more extreme climate change. So what is interesting is that studies have been showing that in overall, when we incorporate information on physiology, as like the mechanistic models, or the local climate-driven adaptation, it tends to decrease the filter range loss projections that the studies are showing. So here I bring a couple of examples. In the left, it's a Sky Island salamander, so 3 different species. So we look at the left and right, and the yellow are the suitable areas in the filter. And when they use the biophysical property of the species, then the range loss is way less extreme. And here in the left is another example, focusing on a bird from Europe. And what they've shown is that when the projections were made for the full data set, they recover more extreme type of range loss prediction than when they look at the populations that they identified as like hot or dry adapted. So if by focusing on these hot adapted populations, they recover a less extreme range loss for future climate change. So here, I'm gonna share with you some of the ideas and preliminary results of the project that my group is working on, in which we wanna infer some possible impacts and adaptive responses to climate change across the Amazon Cerrado environmental gradients, natural populations of lizards from these biomes. So we are doing that first, focusing on 3 different species groups with different ecologies. So we are looking at the generalist, a more lowland rainforest specialist species, and a species that is associated with open areas within the Amazon or the Amazon savannas. And we carry a 3-fold goal. So we first conduct traditional biogeography studies to understand the species and population limits, divergence times, and the background or the neutral population structure. And then we also study the demography, but what I'm gonna really focus now is on the approach that we want to identify this genomic base of climate-driven adaptation. And so here is just a schematic representation of what we do. So we start off doing field work and collecting our biological samples, so the tissues, that we need for genomic data collection. And we also conduct this thermal physiology experiments. So we quantify thermal preferences, critical minimum and maximum temperatures, and the locomotor performance under increased temperatures for these lizards. And we study how these traits vary in space, but we also collect genetic data, both Sanger sequence data, regular sequence genes, and genome level data through reduced representation libraries, RED-seq data. And with this data, we investigate the neutral population structure, demography, as I said, and then we are looking at on the genome data, on adaptive genomic analysis in the search for selection signal in the SNPs that we collect. So we want to see if we can recover SNPs that are under temperature selection, for example. And in the end, so that's what we're still working on. And we will integrate these SNPs and the mechanistic, the thermal data under mechanistic modelings to infer their extinction risks and evolutionary rescue. So it's just the tip of the results. We are working on it. So here I bring for one of the species, the forest species that I've shown the models before. And what we found based on 4,000 SNPs was a neutral population structure in 5 large groups, different colors, different populations here. And they seem to be pretty well delimited by the main Amazon basin rivers. But the ecotone seems to be a region where these at least three of these groups can be in touch, can get in contact. So subsequently, we are running the Genome Environmental Association Analysis, or GEA, using 2 sets of different approaches. So the first one is through redundancy analysis that investigate how groups of loci, they covary in response to multiple environmental variables, for multivariate environment. And it's known to be able to detect processes even when the molecular signal, it's sort of weak. And indeed, we recover 185 candidate SNPs under environmental selection for this group, which is quite a lot that we were from the environmental variation that we were investigating. So as false positives can be an issue with this sort of analysis, the GEA, we are using a combined or a conservative approach in which we want to look for congruent SNPs across different methods, SNPs under selection, right, through different methods. So the second method that we are using is called the latent factor mixed models, and it investigates the correlations between the environment and the genetic variation, in that case, one at a time, while simultaneously accounting for what we call the background levels of population structure, or the random factors in the analysis. So when we did that, we recover less SNPs, 30, in opposite to the 185. But the interesting here, when we look at the space, what we would call maybe the adaptive population structure, it has some resemblance with the neutral population structure, but it has some differences as well. So some potential connections between like Eastern populations, but we also believe that this is a case in which these adaptive, this climate adapted SNPs, they seem to really match the distribution of the ecotone in here. So we believe that that's evidence that the ecotone might be a very important area of source of climate-driven adaptation for this system. So of course, we have still a lot of work to do. And among these, we want to explore more variables, including, for example, maximum temperatures, which are known to be important for climate adaptation in several groups. And once we have the set of SNPs that we are still working on, or in other words, the ones that are recovered across methods consistently, then we will move on with the mechanistic ENMs, with the thermal physiology data, looking at the heat-adapted populations. So the goal is to be able to see, I'm almost done, guys. The goal is to see if there are areas or populations that are more sensitive than others, and if in the future, the ones that have this climate-driven adaptation, if they will be recovered as spreading this genetic variation to the others, and then, let's say, rescue them from their higher extinction risk probability that we infer. So the project, it's ongoing, as I said, and we are still working one species at a time, but the idea is to be able to scale up the approach across taxa and increase its power by looking at species with different distributions, different ecologies. So you've probably seen it. It's fairly broad, the distributions of the species that we are looking, but we want to look at more restricted range species as well, and also provide some independent functional validation by doing transcriptomics and also some whole genome sequence for some of these populations, so we can annotate our SNPs back and see, oh, are they really involved in, let's say, heat tolerance? And we want to also look at some synergies between land use or deforestation and climate change, because they are shown to be very important in these scenarios. So in summary, I think that our previous work and some of the preliminary data, they show that the intra-specific variation or the population level variation of thermal traits and their respective vulnerability and the standing climate-driven genetic variation, it's important and can affect the possible responses and extinction risks of these lizards and their populations. So that's very important, because conservation strategy, they should take that into account, the possibility of evolutionary rescue between populations. And this can only be fulfilled if the populations are connected. So it doesn't matter if they are apart. If there is a important genetic variation, but it can't connect with the others, then it's all gone, right? So, and the other things are that we do believe that the winner-losers idea in climate change scenarios, they do apply for the population level as well. Not only different species have different vulnerabilities, but also populations. And this might seem to be a promising approach for large-scale environmental gradients, such as the Amazon and the Cerrado. And we hope to increase the awareness of the links between climate change and biodiversity, because they are problems that are very interconnected. The biodiversity loss, that's the problem, not the biodiversity, but they are linked problems, so the solutions are linked as well. So with that, sorry for the technical problems in the beginning. I want to acknowledge my research group, which is large and diverse and work very closely with me. And of course, lots of collaborators involved in the proposal. So here I highlight some of them. And thank you so much for all of them, my funding agencies, IBS, José, Diniz-Filho, and the great photographers that make the presentation more beautiful. So here is my contact. Thank you very much.
Hey. Thank you very much, Fernanda, for this great talk and for giving us another excellent example of how to integrate these local processes, this population level genetic variation to the emergent patterns, and how it is important to look at intraspecific variation. And thank you for this positive message that maybe the effects of climate change will not be as extreme as we were expecting before. Sure, we still need to keep working on this, but it's good to have this positive message.
Nice. Thanks, Elisa.
And next we will have Thiago Rangel, and he will be talking about the ecology and evolution of biodiversity.
Thank you, Elisa. Again, thanks for the invitation. Let me share my screen. Share screen. I want to share this screen. And I want to move this out of the way. It's again a great pleasure to be among great speakers and to be speaking on behalf of IBS to this great and diverse and very important audience in South America, mostly in South America. We are the hub in Brazil. And today I will share with you some results of a model that it took around 6 to 7 years to build with several iterations, then it spans over 20 years. And the basic idea behind this model is to integrate over scales the ecology and evolution of biodiversity by merging processes that are happening in the short, small, and emerging patterns that evolve to the larger scale. And by large scale, I mean as large as possible. We are very used to look at now global patterns of biodiversity. Here we have global patterns of biodiversity of vascular plants. And not only this is a beautiful pattern with a gradient, very strong spatial gradient, but it's also recurrent in nature. It happens over and over, very different groups or clades, groups of species. And the first to acknowledge, to actually be surprised and to comment that was our hero today, von Humboldt, Alexandre von Humboldt. He did travel through South America. Mostly, his most important field trip was through South America. It took years and years, and he did not, unfortunately, he did not step over Brazilian territory. Because the Portuguese crown at the moment did not want any German citizen fooling around and messing around and gathering information on Brazilian territory. But he did visit several Spanish colonies that are now independent countries, of course, in South America. So here we can see the same pattern for the tetrapods. And when we see recurrent patterns in nature, we immediately, just as von Humboldt did so long ago, we start thinking of common causes or universal causes. But the problem when we start talking about causes is that we don't have replicates and we cannot run experiments. And that's unfortunate because science is good or science is easier when we can run experiments. But we can't, not at the scale, not with only one Earth, not with replicates. Yes, we are unfortunately running a very bad experiment on heating the Earth. We will not have replicates and we do not have a counterfactual to actually tell that. Or to investigate the consequences as it moves. But for the causes of biodiversity patterns that evolve over millions of years, we don't have that. And basically, what I want to show you today is a model that can do that on a computer. We will evolve biodiversity on a computer by using several of the processes that Fernanda and Sydney have discussed here earlier. So everything I'm going to show you today are simulations that emerge from first biological principles, principles that we know to be very reliable, biogeographical principles. And when we build all these processes, all these principles on the computer, and we run virtual species over realistic biogeographical landscapes that evolve over time and are heterogeneous over space, then we can see the emerging patterns from the principles that we discuss in biogeography. So what we will do is to build machines, to build mechanistic models, and discuss what these mechanistic models tell us on the origin of biodiversity, on the origin of, on the ecology and evolution of biodiversity. So here is the beautiful continent of America, the beautiful continent with its nice shape, and as an homage to Humboldt, we can focus on this very high biodiverse area, which is the tropics of South America, and how diverse it is, and with all its idiosyncrasies and its mountains. And we will discuss the importance of the Andes in this major basin, river basin, which is the Amazon basin, and all its impacts and its evolution over time. However, if we want to talk about the emergence of biodiversity, we need to talk about the past. That's necessary. Unfortunately, we don't have a time machine. The best we can do so far is still to use estimates of previous environments. And as paleoclimatology evolves, we can now look further back in time, further and further back in time. At the time that this model was built, the furthest we could look with accurate, or as accurate as possible, estimates of precipitation and temperature was 800,000 years. First, I must start saying that it's not enough. It's a work in progress. We can now move back or see back 5 million years. So maybe an update of this research is on the process. But with 800,000 years, we can get most of the recent fluctuations in climate change and see what patterns emerge from these fluctuations over time. So just as an illustration, if we can get a single location in South America, and we can get its maximum and minimum temperature, its maximum and minimum precipitation, we can represent those values as a cross in this two-dimensional environmental space of temperature in the y-axis and precipitation in the x-axis. And then we can represent all the locations in South America and see how it changes over time. So what you see now is the beating heart of a dynamic climate that evolves over time. And that magnitude of change, those oscillations, those glaciations, they have to cause a huge impact on the biogeography of biodiversity, on the species that inhabit the area, the domain of South America. So what are the consequences? How much can we tell from the first principles on the effects of this climate change on the distribution of biodiversity over space and time? So imagine a virtual species. We will assign to this species this niche, this climate tolerance, and by assigning this niche, we can immediately assign the possibility of this potential niche in this potential range in geographic space, because there is an interchange, a duality with niche in the environmental space and distribution in the geographic space. Where else can it be? It can be anywhere else if the species can disperse, can colonize, can jump over unsuitable area. And so we can implement that. However, if the species cannot move over areas and there is an isolated population, we assume there is no gene flow, And by not having gene flow, there is a potential for independent evolution. There is inheritability of a niche. And then if there is climate change, what may happen is that part of the range may be lost by climate change. So the temperature or the precipitation no longer is suitable for the existence of the species. However, the same climate change can also create a new suitable habitat for the species to expand its range. So this is also included in the model, and we can also allow rescue by migration within the range of dispersal capacity of the species. So by saying that when a range breaks into two fragments, there is inheritance of niche, we can start building into the model a process of evolution. And if there is enough time in isolation, these two populations that could, in the past, interbreed, by providing enough time in isolation, these two populations may start to be called a different species. Of course, this time for speciation is better measured in generations. I'll talk about this briefly. And the number of generations vary from from species to species. So if there is enough time, we can then first maybe see different subspecies, and if these subspecies can further isolate, some of these subspecies may actually become extinct. But if over time this isolation remains and preventing gene flow, then we can say that these two species, these two populations become independent species. And so here we have a parameter that indicates how much time it will take for two populations to become independent species. And here I'm just representing an origin, an ancestor species, and its two independent populations that have evolved. So no longer we can see gene flow between these two groups, the groups on the top and the groups on the bottom, although we can still see gene flow between subpopulations that belong to the same species. So now we have a very sophisticated model that integrate biogeographical processes that have been documented and discussed in the literature over decades. But now, with the possibility that the simulations model provide, we can integrate this process into a single framework, and then not only evaluate how these processes combine and what patterns emerge, but we can also estimate, we can gauge, we can measure the importance of independent process, of each independent process over this very realistic dynamic landscape. So here, for example, I will show you a single ancestor. Here is a Indian ancestor that starts occupying this area that is here shown in blue. And here is the niche of this single ancestor. And then I will play the tape of biogeographical evolution for this species using these principles that I have shown, and see how multiple species emerge from this process. So here you see the first sign of fragmentation. This population here becomes isolated. And then what you see here is this very small niche for this very small population. It's still not independent because not enough elapsed time have passed. So it's not a speciation yet. But if you just let it go for long enough, you will see populations popping up and disappearing. And then one day you will see speciation, a group of subpopulations that have remained isolated for long enough to become an independent species, so that no further gene flow becomes possible. And then we can document all these principles in this simulated environment, so that we can study what conditions cause speciation and extinction processes. So now we see multiple species piling up or accumulating. So we have two here. And so the colors that changing over this map indicate species richness. And then you can gradually see species richness accumulating along the Atlantic coast. And then you can also gradually see species richness accumulating along the slopes of the Andes. So, and that's for a single ancestor. What would happen if we let this, this process run for long enough? In other words, how realistic is the model? Well, if we look at the observed patterns of species richness for birds and mammals, then we can compare this against the simulated. And here is an astonishing pattern, a similarity among patterns. This is simulated just using first principles. It's not using real data, real biodiversity data. We are generating, we are creating biodiversity that looks very much like the observed ones. In fact, we get this R squared of over 65 for using an Indian founder. Here are the residuals. The residuals are somewhat dispersed or scattered. There's still a lot to improve in this model, and we hope to do this over for future iterations. So here again, the pattern of species richness, and here's the simulated. This is how similar they look. This is how it's interesting how the Andes emerge here in the simulated pattern, just as they exist in the real world. And again, this is just using 800,000 years. So we believe there's something, and I will speak further about this. We believe the characteristics of the continent and any dynamic of climate change has the potential to generate similar patterns of species richness. Again, here's for plants. Look how similar it is over the slopes of the Andes. Here's in Ecuador and Colombia and Venezuela. By having a simulation model, we can also do some experimental biogeography, which is something that we cannot do in the real world. We can change dispersal capacity of the virtual species to see how the patterns change. We can change evolutionary rate. We can change time for speciation. We can play with parameters. We can change intensity of competition. We can change the origin, the ancestor, the distribution of the ancestor species. And that's again for von Humboldt, we can change topography. Von Humboldt has discussed so much about the importance of mountains, and he did visit the Andes over and over again over years, and he talked about the importance of the Andes. Here we can now test most of his ideas by bulldozing the Andes, for example. We can run a counterfactual test of the importance of the Andes by removing it from the simulation, just to check if biodiversity emerges in the same way. We can transform the Andes into this flat grassland and see if patterns emerge. So here are different alternative topographies for South America, using a mathematical kernel to smooth area, the topography over South America. And what we see is that niche evolution, the speed of niche evolution, the distribution of the founder species, and the interaction between topography and climate heterogeneity are the most important factors explaining the distribution of biodiversity in South America. What else does this model tell us that we cannot observe in nature? So if all these assumptions are relatively realistic, if they are useful, what else does the model tells us? Well, imagine a single ancestral population, just one initial species. So the range fluctuates over time, and then suppose a range fragmentation. during this period in which the species is mostly changing the size and shape of the range, but not exactly in the process of speciation or extinction, we call this the persistence trajectory. After range fragmentation, we will call this a speciation trajectory. And for those species that go extinct, for those populations that go extinct, we can call this the extinction trajectory. So here we are basically splitting the lifetime of the biogeographical lifetime of a species into 3 different stages: persistence, speciation, and extinction. And then we can draw analogies from the art of photography and use this old technique to photograph into a single frame movement. How is it done? So here we see that the brighter the shade, the more overlap there was of this object, in this case, this woman, over different clicks, over different shutters, activations for the camera. So the persistence trajectory for us is a museum, the speciation trajectory for us is a cradle, and the extinction trajectory is a grave. So the more we see species persisting in a region of the map, the more museum that area is. The more we see species speciating in an area, the more cradle that area is. And finally, the more we see species going extinct in an area, we can call that area as a grave, a place where most species go extinct, where it's frequent for species to go extinct. And by decomposing the lifetime, the biogeographical lifetime of a species into these 3 categories, we can then partition the total species richness over these components. So what we see here is that the evolution of these patterns. We see here museum species richness piling up or accumulating. We see here the cradles, the map of cradles where most speciations have happened. And we see here where most species have gone extinction, have gone through the process of extinction. And what we find over the simulation is that it's very common for cradle graves in museums to be in the exact same place. For example, the Andes is simultaneously a museum and also a cradle. This is not a dichotomy. They can overlap. And also, it's also a grave. So we see as climate move up and down and climate shift, we see species going extinct at the top of the Andes by biotic attrition, as Rob Caldwell would say. So here are the maps of cradles, the graves, and diversification in museums for species richness in South America. With that, I must acknowledge and thank all my collaborators. This is not a single-man project. It took years and years of work to build this project, this simulation model, and a very fruitful collaboration with paleoclimatologists, especially Neil and Phil from the UK, but also Alexandre, Marco Tullio, Fernanda, Karsten, Rob, and Will, a paleobotanist in the Netherlands. I also must acknowledge and thank my the institutions that have participated and all the support provided to this project. And with that, I thank you very much for watching. And today is a day to celebrate the legacy of von Humboldt. Thank you very much.
Thank you, Thiago. It was a great talk. I think we can invite everyone now to go back. Sydney and Fernanda is going back. And before we start the next section of this symposium and start to discuss and to have the questions from the audience, I would like to invite my good colleague, Miguel Matias from the University of Évora, that is one of the organizers of the overall idea of the Humboldt Day. Welcome, Miguel. Thank you for coming.
Obrigado, Alexandre. Thank you, everyone that is watching. My name is Miguel Matias. I'm here on behalf of the International Biogeography Society, and I would just like to thank Elisa and Alexandre for putting this event together. Alexandre said yes even before we had many events confirmed, so thanks a lot. But also to the three amazing speakers, Sid, Fernanda and Thiago. The research was amazing and it's really interesting. We put together this event at a time where traveling for conferences is extremely hard and there's lots of uncertainty. So the point was to basically connect people, ecologists and biogeographers from all around the world. We had events from New Zealand to Madagascar, from Norway to Colombia. So we would like to thank you all for watching. We just came from Mexico for an event in Mexico, and the next one will be in Colombia. So keep watching these events, and we thank you for that. Thanks.
Thank you. And congratulations for all the effort to put everything together.
No worries.
Okay. Oh, okay. We can go now to the questions. I think we have several questions from the audience. And before starting the questions, right, Lisa, I think that we have this overall idea now that I already mentioned about we always try to understand what's going on at macro scale by thinking in process at a more local scale. And when macroecology started, let's say 20, 30 years ago, we are basically documenting the patterns, and it was very difficult to get the data. Today it's much easier to get the data. And now we can focus on process, and before we have all this discussion about different processes giving or origin to the same pattern, all the under-determination problem of different possibilities, and now we can try to disentangle some of the process and see how they will generate the pattern at broad scale. But even so, as Thiago said, and also see it about the models, our mechanistic models are still too simple, or at least they are not able to incorporate, not because of the models, but basically because of our limitations in terms of data of natural history of different species. And I think Fernanda mentioned this idea of trying to parameterize the models for different species, or at least for different groups of species that will have different natural history, or they will respond differently to the same environment. Although even so, I think that we are advancing very fast in terms of this idea of better linking patterns and process in macroecology and in biogeography. Okay, Elisa, let's start to the questions, or you want to do some overall question too?
I think you summarized it quite well. Yeah. We can go to the question.
Okay. Well, let's see. We have, of course, we have about 160 people watching, fluctuating a bit, but we have now a bit more than 100 people in watching this in YouTube and Facebook. And we have, of course, lots of questions here and lots of compliments about the very nice talks that we have. And I think we can start with José Dazi, that was our PhD student. And let me see how this. Okay, here we go. So everyone can see the question. Dazi is asking about process related to gradualism or punctuated equilibrium, especially when related to neotropical biodiversity. And so that's the first question. Who wants to
it continues, answer? Right?
It continues.
So, yeah,
let me see.
The next one.
Okay. Sorry. It's correct to think that some traits such as range follow gradualism and other traits such as food type will follow punctuated equilibrium.
I guess maybe Thiago could take this one. I agree with you. We don't see that much, right, in the literature, specifically for neotropics. But I don't know, because I think that Thiago models some of this more long distance or more abrupt type of range shifts in the models, right? So maybe.
Yeah, well, I would not dare providing a permanent answer to the question. It's more like a research agenda than a question. But anyway, with the kind of models that we have been working with, which basically integrate ENMs with dynamic landscapes and some known biogeographic process, we do see a mix of these processes over time. It depends very much on the dynamics that it's actually going in the landscape over time. We can see bursts of speciation, but we can also see a gradual increase, a permanent increase with gradual steps. So I don't like the dichotomy of having to dissect between either one or the other. I don't think it's useful. I like to think on the processes that will generate such patterns, and then discuss how these processes operate and what are the factors that contribute to the emergence of speciation and extinctions.
And I think one interesting issue with this punctuated equilibrium question that Idazi asked us is, Thiago, is perhaps we can use the model, for example, to try to understand this, because one of the great discussions about punctuated equilibrium is if stasis is a consequence of environmental stability or of some intrinsic components of the species or some other constraints, for example. So actually, we can have some kind of if we can try to disentangle gradual continuous evolution responding to climate versus a more static dynamic independent of climate change, and if you can compare this with the real data, it would be a nice idea, right? But I think that it does is right. We cannot see lots of this discussion. Mainly the discussion about punctuated equilibrium comes from paleontology or paleobiology literature, and not geography. I think perhaps we can have some links. That's very nice.
Yep, true.
Well, we have lots of other questions. We have a question for Fernanda. Let me put here. Okay, here we go, Fernanda.
So that's, hi, Gabi, thanks for the question. I'm going to read if someone hasn't. So she asked us if would it you'll also be able to take into account different dispersal capacities among species in the mechanistic ENMs. So thanks for the question. It's very interesting. I guess that maybe individually, because usually the ENMs, they are run for each species at a time, right? So at least on the approach we are using, the dispersal or the movement of individuals in the space, we usually use like more of resistance surfaces afterwards, when we are trying to, for example, project in the future, the last step where we are still working on, so I don't have results on this yet, but so the different dispersal capacities, they weren't incorporated in the model in the beginning. So what I've been thinking more, for example, even for landscape change, deforestation and stuff like that, for example, it's more like to subtract the surfaces after I have them estimated for using the thermal data. Then you can look at, okay, these are the potential areas in the future, but how is the landscape there? So if there is a land use or deforestation, then we use like resistant surfaces to look at the evolutionary rescue, discounting these harsher, let's say, environments. But we haven't used this as a parameter for ENMs. That might be possible. I don't know if my colleagues have something to add on this, on the model development aspect of it, but I agree that when we want to look at on a comparative level, looking at different species for one single map, let's say, if I want to have an adaptive potential map of lizards in South America, then the different dispersal capacities, they need to be taken into account. But so far, we are not using in the ENMs yet, but it's interesting.
Okay.
Fernanda, can I jump in and
sure, please
make a related question to you? Yeah, I was wondering if it would be interesting and possible to include demography in those mechanistic models.
Based from genomic data, you mean like effective population sizes and yes, it's absolutely possible. Some other approaches, they use, for example, demography from empirical data to simulate or to restrict the carrying capacity of, so let's say, so there are some studies actually in Brazil, in the Atlantic forest that uses that even for lizards. So he integrates, it was a study from Ivan Prates. So he used demography as the empirical parameters from demography as carrying capacity for each cell or each pixel. And then you can project how many individuals this pixel can carry in the filter, let's say, on the genome variation in the filter. So it is possible.
Great, thank you.
Thank you.
Okay, we have two related questions. First one is by Tamara. It's also to Fernanda about phenotypic plasticity, incorporating phenotypic plasticity, and she's saying that in a following, she's saying that it would be much faster than expecting for genetic changes, right?
Yeah, there is the other one, right, from Hosanna.
Yeah, and also Hosanna commented that, hi Hosanna, that about the importance of phenotypic plasticity, right?
Sorry.
No,
so I think these questions are very, very important. And as I'm not sure who did respond from the ENCT in the chat, I completely agree. That's the frontier of how to include the portion. It was you, right? Yeah, I suppose so. And please help me on explaining that. So the portion of the variation that comes from plasticity or epigenetics into these models. But the thing, but I would say that for us, for me, my group, that we are looking at empirical data at the population level. So that's something almost virtually impossible to quantify how phenotypic plasticity change across, let's say, 200 localities that we sample. So I guess that this could be a parameter for the species. That's what I understand what José Diniz answered from the ecography paper. So at that study, please correct me if I'm wrong, it was like a theoretical parameter into the model, and it was for the species as a whole. And when the phenotypic plasticity was considered, the possibility of evolutionary rescue was even higher. So it's important. It's certainly an important process. I would just say that for whoever is working with empirical data, it's a huge challenge because we are covering a large area. So we need to sort of use, it is a, let's say, less than ideal approach in the sense of we have some processes left out. But if we were to consider these things, we couldn't have this geographic coverage. So that's kind of the trade-off of the framework we are using right now. So we want to look at geographic coverage because we want to see how these things change in space, in this gradient. But later on, our idea is to do more focused sampling and do other sorts of genomic approach that could help us include plasticity, phenotypic plasticity. But that wouldn't be feasible with this dense sampling. Hundreds and hundreds of samples, and actually things that weren't collected 5-10 years ago. There are some samples that are from museums, and so that's the sort of limitation we have for this type of coverage when you have empirical data. So I guess that's the big challenge. It's how can we, and that's something that I would love to hear Sid and Thiago and José as well. So how can we cross-validate the models, the empirical, the theoretical models, with a good interest-specific or population type of variation? Because it's hard to do that, money, time, all sorts of constraints. So I would love to hear, like, this interest-specific type of variation, how could they be incorporated in these theoretical models? Thanks for the question. Really important points.
And Idazi also is adding a bit more in terms of epigenetic effects, right? So I think all these are frontiers in terms of not new, but different forms of inheritance at population level that could be escalated to a broader scale. And I agree with you, Fernanda, it's very difficult. And as I said in the chat, it's very difficult to try to parameterize the phenotypic plasticity, not the environmental variation, this is random variation, right? But the coupled interaction between the environment and the shift in terms of morphology or anything that we are trying to model. It's very complicated. I think it's very different, and we do not have lots of experimental and empirical data to calibrate this. Do you want to add something, Sid or Thiago? And okay, let's move on. Idazi is also doing some comments and about limits of evolution, and I think it's quite similar, Thiago, to another question by Marcos, perhaps Marcos Dantas Queiroz. Perhaps you want to answer both together, right? About secondary contact or hybridization zones or events during the simulations.
Well, thank you for both for the comments. Thank you, Idazi, for the comments. Indeed, you have seen earlier and earlier, like baby versions of the model. I guess most people in the department have heard me talking about the basics of this model. But then, of course, there is a lot else to do. Nature is not as simple as our models are. But I also like the incremental steps in which we learn from previous iterations of the models, and then we feel confident to build new ones and improve here and there based on empirical evidence. Then Marcos mentioned the possibility of incorporating secondary contact and hybridization. Yes, this is something that we have been working on right now. So speciation is not an event, it's a process. And I like to repeat that all the time that I think of my models. So at this moment, it's just like a threshold in time, and it just happens instantaneously. That's not realistic. It may be good enough to capture some patterns, but that's not how speciation actually happens. So we are now working on a future version of this model and a next iteration, in which gene flow is a gradual process. It's dynamic, and and it may happen, and it may increase or decrease with more or less contact. So under this population genetic version of this model, we will have secondary contact and hybridization happening as a natural process, and continuously over time, we will have all possible degrees of evolutionary separation between populations. So yes, I completely agree, and the group believes that it's the way to go forward, especially if we want to get the high biodiversity patterns along the Amazon basin, not only along the Andes, but also within and along the Amazon River and within the Amazon basin.
I think we have a question by Isadora. Thiago, you can go
Yep.
on, keep going with the following the reasoning.
It seems that considering the Amazonian founder, a lot less grave areas than the founders. So Isadora, given the the paleoclimate variables that we have used, which is just temperature and precipitation, the Amazon basin in the model becomes almost like a flatland. So the seasonality is relatively homogeneous over space for both temperature and precipitation because of the scale, the spatial scale of our analysis, and because the Amazon basin is a flatland. It's just topographically flat. And also, we have not included dispersal barriers that the rivers can cause for some species. The next iteration of the model, the one that I was talking about just a second ago, when answering to Marcus, is intended to improve or become more realistic with regard to these processes. We do know that major rivers play a role as a barrier for several species that don't have the capacity to disperse over rivers. And so we think that the problem of capturing patterns within the Amazon basin is still to be solved and interconnected as a biogeographical processes to these macro processes that we have already incorporated. So I could not agree more. It's a place where there is probably a lot of speciation, there's probably a lot of extinction, and most likely the model is missing all that.
And Thiago, sorry for interrupting. So, but do you have the plans to do that? Include the barriers, and if you would do that, how would that be? Like more like a fixed barrier or a temporal? Is that a way to include the barrier in the model and a dynamic barrier? Because there is still this old discussion, or when did these rivers establish? And even after the establishment, there is a lot of dynamics in rivers, recent dynamics. So how is the river system coming on the future developments?
It's a work in progress. We are working on this. And we are collaborating with geologists to provide an erosion model, a paleo erosion model, and a model of how rivers are dynamic over space and time. So the origin of the major rivers in the Amazon basin and their change over time with the uplift of the Andes, going back possibly as further back in time, or even further back than 5 million years. That's the plan. It's a work in progress. It's yet another link of interdisciplinary collaboration to build these big models. And it's not something that we can finish this year or next year maybe. But yes, we are planning to do that and to incorporate the hydrodynamics of major Amazonian rivers, not only Amazonian, but South American rivers into the model, and the formation and disappearance of barriers over time.
Nice.
Oh, that's amazing. Can I just chime
Sure.
in and have a question for Thiago? So, do you have any expectation regarding the emergent patterns you will find in this new model? Because you're adding a whole new layer and going like a level, like a finer level of genetic variation and gene flow. Do you expect to find species richness patterns that match the actual ones even better than the ones you found before?
I think I will be very happy to keep the same pattern of fit between model predictions and real world patterns. Let's say above 65% fit, which is already good enough. But what I will mostly be interested is to be able to find that for some groups of species, the dispersal barriers are more important than for other groups of species. So I will be most happy to be able to have models that are clade specific with regard to the biology and natural history of these clades, instead of this very super general model that just capture patterns that are so broad that basically capture patterns that work for most clades. So if this plan works and if we get the process and the model right, we will be able to try to incorporate a little bit more biology and clade specificity into the model to be able to say that, well, dispersal barriers are important to the diversification process of this clade, but not that one because of this or that.
And I think this is interesting, Thiago, because we always talk about incorporating, for example, phenotypic diversity in terms of, for example, for a large organism, a river may be not a barrier, but for a small organism, it works. So it links perhaps with something that Sid is doing, although it's not in terms of the biogeography, which leads us to the question by our friend Frederico Falero that asked something to Sid about the speciation and the models in terms of body size.
Okay, thank you, Fred, for the question. Actually, the model is not an evolutionary model. We don't have, different from Thiago's model, we don't have a special structure. We don't have an evolutionary relationship. It's just the fit that emerged from a thing that is shaped like a lizard, that exchange heat and water like a lizard, and given the microclimate, these changes happen, and the end of the day, you have a distribution of probability. What is the body temperature, final body temperature of this large population? So the measure is actually a fit of this thing that is a hypothetical lizard to the climate, to the microclimate that decomposes the macroclimate in small scales and in time. So it's just an emergent pattern that comes from the bow plane, the body plane of that thing we call lizard.
Okay, and I think we have time for two other questions. Keep going with the speciation idea. I think, let me find here.
Just to mention one more aspect of Fred's question. He mentioned that maybe the model would be better for predicting species that occur in open areas. Just to try to synthesize how the model works. In this environment, what this animal is, we set different habitat cover or shade covers. There's shade, there's sunlit area, and there's 50 sunlight and shade. So the animal can change these microhabitats depending on how the temperature, the body temperature is. If it's too warm, he can move to a shaded area or can move the exposed area, and then it adjusts its body temperature according to these dynamics. So not necessarily it works better for open areas, because we can actually set the shade cover to a full shade cover. But in the model, the mechanistic, the microclimatic models, they are those from Kearney, Michael Kearney, microclimate. It decompose and make the calculation, physical calculation of temperature in water, evaporation, everything that happens on the ground, on a shaded area, on a sun's exposed area. So it can work in all these types of environment. What we were working on was to inform shade cover that resembles best the vegetation cover. We can simulate or emulate the vegetation cover and inform what is the proportion of shaded and exposed area in a particular landscape, and model this system in this particular landscape.
If I may ask Sid a question, I would like to ask if you have explored, or if you believe for the sort of models you are using, the climatic variation. So what is the expected? I mean, we've seen a lot of new layers of microclimates that are derived from global layers, or you could go and validate them in the field. So what do you expect would be the effect for the sort of mechanistic models you are using if you were to use microclimatic environmental data?
I have no idea. Actually, this is a great question. Actually, it is.
Did Sid?
Sid travou. Voltou.
Okay.
I've discussed some of these things with some colleagues in an attempt to validate those microclimatic models with real data. The groups that has developed those microclimatic models, they have made such a validation, and apparently it works pretty well. So I don't really know the answer, if it's better or it's not, it fits or it matches or not the data taken from the field. Something that we could do, and these collaborators are doing actually, is to install data loggers in AGA models in the field to monitor the body temperature and the evaporation in the model, so they can calibrate these microclimatic models with this empirical data. But this is all I think we have, or all I think there is around.
Okay, I think we have two general questions, and I'm going to put. I think that we can end the symposium and with these two very general questions. In the meantime, just a sec. Just showing in the screen an opinion by Gabriela Zucchini about Thiago's comments on the Amazon idea. But the questions, I think that we have two general questions, and perhaps you guys can comment these two questions, and so we can end the symposium. The first one by Marina. I don't know, Marina. She's saying about, what do you know about the geography of speciation in the Neotropics? Is speciation majority allopathic or not? It's a very general question.
It's a pretty good one to wrap it up, right? It's pretty general. But it's hard, as I would say that, as Thiago said in the beginning, I don't think it's very useful to go too much into the dichotomy if it's majorly allopathic, but I would say that definitely new studies, as we are using this local level, both biological and environmental data, they've been showing more and more examples of non-allopathic speciation, because it's being considered majority allopathic because sort of the easier, or not easy, but I mean the more empirically easier way. It's hard to use the word, not use easier, but it's easier to understand allopathic speciation. But as we are gaining more and more data, I think that there are more and more examples of ecological speciation, sympatric speciation across the Neotropics, not only Amazon or. So I guess as we gain more and more momentum on these approaches that links the micro and microevolutionary and macroevolutionary process, I think we will see more and more examples of ecological speciation. That's my take on the view.
Yeah, just to add my 2 cents, I agree. I absolutely agree. There is a major intellectual impact of the idea of allopathic speciation because it makes sense with a lot of other major processes over time and space. And we see range disruption or range breaks among clades, and then we infer allopathic speciation. So it's easier to think of allopathic speciation. But the more we have genetic studies, such as the ones that Fernanda do, look at phylogeography of clades, then we can actually see that this broad pattern that we can look from a macroecological perspective does not really exist in a more local scale when we start talking about species that are similar or evolutionarily related within a regional scale versus a continental scale. So I think the question of is it mostly allopathic versus all other forms of speciation, the answer is structured over scales, spatial scales and temporal scales. And if the further back in time and the broader that we look in the domain, the easier it is to see allopathic speciation. And the reverse is also true in the other direction. So the more data we gather over space, and for a more detailed phylogeographic perspective of the neotropic biodiversity, the more we will see examples of non-allopathic speciation at the regional to the local scale. That's my 2 cents to this question. I think they all exist at that same time. It's just a matter of what lens you are using to look at it.
Okay. Sid, do you want to add something? Okay.
No, I'm okay. I think I'm the less capable to talking about this in this group. But if I may add something based on our results, what actually if we accept the idea that the sweetest or the most suitable climate has more species, what this would be telling us is that a particular climate that suits more or for a given size class, it enables to accumulate more species of that size class. So so if we accept this model as fairly good or predicting fairly well this distribution, I would say that specific conditions allow for accumulation in the same area of species, similar species, with the same range of traits.
Mm-hmm. Right. Okay, I think we can go to our last question, although it's directed in principle to Thiago. I think it's a very general one, actually. Ricardo Ieda asked about future climate change and how our models, although we are usually, in all three talks, we are talking about reconstructing historical event, and although Fernanda mentioned the response in terms of the rescue, I think that idea of coupling local process and broad-scale patterns would be very interesting in terms of predicting what's going on in future climate change, right?
Well, I'll just provide one sentence and then pass the microphone to Fernanda. So we do incorporate in the model climate change, past climate change. We did not run the model to the future, and the reason is that we are looking at an evolutionary time scale, like the speciation scale. Every step in our model is 500 years. And we are now moving to 1,000 years so that we can look back 5 million years. So we are interested in these deeper and deeper scales. And the processes that we are incorporating into the model are not the same that we will need to understand the impacts of current or anthropogenic climate change over the biodiversity that we know. So the model is not designed for that. For that, you need to talk here below to Fernanda. She will have the answer.
Yeah, no, I agree. I mean, it's a matter of different time scales that we are looking. So far, I guess, for projections, unfortunately, we are most looking at species loss than species gain, right? Because it's just the models, they are driven, like Thiago's models, they are driven across thousands or millions of years. And whatever projection we are doing, it's for the future is like a few decades, right? So I think that these models, they can bring us a lot of insight about the range of species in the future, the projections, I mean, the species range and species loss, unfortunately. So it's a bit hard to project how speciation or species origin would be taken into account in these scenarios. But absolutely, I guess the changing of patterns, which is his question, that's what we are trying to do, right? Play a little bit of a crystal ball, which is, it's very hard to falsify as well. So it's something that it's us to bring awareness to the question, but it's not something we can actually replay, or, I mean, hopefully, I usually, I don't know if there will be someone in my grandkids that will actually sometime in the future look and validate or not this projection. So it's a little bit of a time machine trying to. So I guess it's mostly mostly in these two things, right? The ranges of the species and species loss. But I think that's a very important frontier, because we are looking at neotropics, the huge and amazing biodiversity that took thousands and millions of years to build up, but to lose it, it can be way faster than that. So at our own time scale. So I guess it's a bit of a challenge and a frontier, how we could project there even further away.
Okay. Well, I think that's it. I think, Elisa, what's your final words or ideas?
No, I just want to Thank you for the opportunity to have this great discussion and to be among this great group of researchers.
Pleasure. Well, once again, I would like to thank to International Biogeography Society, IBS, for the opportunity to organize this event here in Brazil. It was great, and Miguel was here to see what's going on. It's a worldwide effort to do a homage to Humboldt, and in some way to put all the biogeographers and macroecologists in contact to discuss all these issues. And so, in the sense, I would like to thank you again, Fernanda, Thiago, and Sydney, for the talks. Fantastic. We can keep discussing this. And also, in our National Institute for Science and Technology, Fernanda, in the ENCT, one of the ideas is exactly this kind of idea of putting different things in different scales and different processes together, as Sid is doing, as Thiago is doing, and it would be very nice to discuss more with you about your project and how we could join and work together on all these topics, right? And so
Absolutely.
I think
it's a pleasure.
Okay.
Thank you so much. I would like to take just 10 seconds to again, thanks. I had a great time discussing with you all and meeting you as we
new times.
Yeah.
Okay, thank you very much, and see you soon. Hope.
Sorry.
Bye. Thank you.
Bye bye.